We are here at Sorbonne University in Paris for the 2018 annual meeting of the ITFOC, Information Technology Future of Cancer project, where breakthrough approaches in oncology are presented. The ITFOC takes precision medicine to a whole new level, predicting patient outcomes to treatment according to molecular data by using innovative computational tools. Cancers are not all the same. Nowadays, we can characterize cancers regardless of where they originate in the body, but according to the molecular makeup, by looking inside the cell. If a few years ago we were able to sequence the human DNA in a few days, now we can do that in an hour or two. This means huge amount of data encoded in the human DNA. What is truly personalized medicine? The idea of targeting specific anomalies in cancer cells is not new. However, our understanding of the disease is getting better, and so are the technological advances. We have discussed with Nora Benabils, ITFOC project coordinator from the French Atomic Energy Commission, about the vision behind the project.
So it's my pleasure to close the workshop and the annual meeting. We had, I think, a very productive meeting all together with the people that we invited yesterday and also the consortium partners of the IT Future of Cancer. So yesterday we had a lot of discussion about the type of data we may use for the modeling and also for the contest we would like to organize at the end of the year, and also regarding the validation strategy we may have for our models. So it's a very tricky issue to get the access into the hospitals to have access to data. So how could we do that, especially in the part of the contest that we were organizing at the end of the year. And so we discussed also with the LNE, so the Laboratoire National de Métrologie et d'Essai, how we should organize this evaluation meeting, big evaluation of the sensitivity and specificity of the model and how well we could predict, for instance, the drug response for a patient, so to be more specific, more sensitive. So this was another strong discussion we had. And we also discussed in the afternoon with the clinicians. So what are already today, by instance, like in the Desiree cohort, and how the decision-making process is done. And so using additional information than the regular clinical data. And what type of performance do they have on breast cancer. And then we discussed also with the surgeon, specialist in breast cancer, so Charlotte Ngo, who explained us her point of view as a surgeon on how this type of methods could be used and when in the process of having a patient coming and to have a diagnosis and then to have the right prediction, the right prescription of a treatment. And so then yesterday evening we had the last presentation on the plan génomique in France to have an idea of what's going on, at least in the context of the French genomic, and there is a lot going on. And so how we can contribute, if possible, if needed, to these different plans. Today was the annual meeting, and I think we had also a lot of productive discussion on what was done this year, a huge work, and with some very nice results on the mechanistic part, but also on other modeling sections, also on the data specification, and at the same time on how we should organize our context. So I would like to thank warmly the person that attended the meetings, the consortium partners, but also the people we invited, Brigitte Serroussi, Romuald Gorgil from the LNE, and also Charlotte Ngo, Christophe Calvin. And so it was really great to benefit from their presence and their expertise. And the very nice dinner we had yesterday also to discuss with all the other invitees that joined us afterwards. And we look forward to have a successful contest organized at the end of the year, and also to have nice results to be presented to the clinicians and for the benefit of the patients. Thank you very much.
Hans Lera from Max Planck Institute for Molecular Genetics explains the main directions of the ITFOC project.
ITFOC is a continuation project of a flagship application which we originally put in the last call, and a part of the development of a future project, Digital Twins, in which we try to solve the key problem of current medicine, the fact that the patients do not respond very often to the drugs which they receive. So we are in drug therapies and drug-based prevention ignoring one of the basic problems of medicine, that all humans are different. We have different genomes, we have different diseases which are superficially similar, and this difference has to be taken care of in any therapy. That is the case in surgery, but it's not the case in any drug therapy, because drugs interfere with molecular networks which are different from individual to individual. So in ITFOC we are trying to use breast cancer as an example to develop personalized therapies for this tumor form by characterizing the tumors and the patients in much more depth than has been done before, and then to develop prediction algorithms, in particular mechanistic models, which are able to predict out of this deep molecular analysis the effects and side effects of all possible drugs which could be used on the tumor. So it is a relatively small project, but it is embedded in a much larger concept of trying to truly personalize therapy, prevention and well-being measures in all different disease areas, not just breast cancer, not just cancer, but all different medical areas, prevention and well-being.
We have discussed with José Fernandez, expert at Marie Curie Institute, about the role of big data in the fight against cancer.
IT for Cancer, it's giving us the opportunity to challenge some of the problems that we are facing today in cancer research. Hospitals are gathering great amounts of data about patients, how patients respond to treatment, and we are in the unique situation of having enough computer power to be able to analyze that data. We have growing amounts of data, and we have the algorithms which allows us to run the analysis of those large collections of data. Some of the problems that we are facing, for example, are when it comes to shared data. It is complicated when it comes to shared data across a number of hospitals, because every hospital has a duty to keep the data of their patients secure, and identifiable data cannot be shared. We have a number of national and European regulations on that sense. But there are ways of sharing information without sharing the data, and that's some of the approaches that we are taking in IT for Cancer. There are ways of creating synthetic data sets which represent real data, but they are not real data, but can be good enough to test different algorithms and train computer models. We have different approaches when it comes to extract information from the text. And again, here we have new algorithms which allows us to do this. Because at the end of the day, right now we are treating patients with information that is being acquired in the past. The only difference with big data and artificial intelligence is that things can go faster and we can analyze more data. But there is no many changes in this approach since Hippocrates and in modern medicine. You know, you always use the same approaches. You treat your patients today based on the findings from patients from last year or a few years ago. New ways of analyzing data as well allows us to find similar patients, you know, in a way that if you have information, nowadays we can get information, genomic information about a particular patient, perhaps there is no much information about the mutations that were found, but if we look into historical data, perhaps we can find other patients with similar mutations, and in a way that we can predict the response of those patients. And this is the kind of approaches that we are undertaking in the project. Cancer is a disease of information, so that it's very amenable to this kind of approaches. We have lots of information in terms of genomic data, we have radiology, we have pathology data, so that we can use artificial intelligence approaches to analyze data in a way that we can stratify patients. We can identify patients which will behave in a similar way based on their pathology responses, you know, the data that we get from the biopsies, or based on the information we get from the MRI scans or the mammographies. For this, cancer is pretty unique in a way that we can use these information to inform physicians and to give the best treatment to our patients. And this is, you know, in a nutshell, this is the pilot approach within the consortium of IT for Cancer. We're focusing on cancer. Within cancer, we have chosen breast cancer. And within breast cancer, we are working on triple-negative breast cancers, because are the ones for which there are no good treatments right now. And we hope that these approaches will allow us to find better ways of treating these patients.
My name is Felix Dorea. I work as a bioinformatician at Alacris Theranostics in Berlin, Germany. And we are in the ITFOC project responsible for the data integration and data storage. So we connect all the different omics data, clinical data that will be generated during the course of the project, like exome, transcriptome, metabolome data sets. And we make sure to connect all these data in a meaningful way based on the clinical questions that should be answered. For example, which treatment can be predicted as a personalized treatment for a given patient. So I think this is the strength of this project, that it has various different data types that can be used as input for the models, for different models, mechanistic models and constraint-based models, machine learning, so different computational approaches that are being applied to the data and can then be used to come up with new therapy options for patients. So I think this is the best or the specific strength of the project, to integrate different types of data and also different modeling approaches to find new treatments. So I hope we will find very interesting results during the next years. There are many challenges, of course, regarding data integration, especially with large data sets like whole genome, whole transcriptome, whole exome and whole metabolome approaches. So, of course, one challenge is to the sheer amount of data, but then also the heterogeneity of the data. So we have to make sure that we use standards that are also able to exchange the data with other users, for example. So, for example, we use SBML as a community applied standard for models, or other standard data types for the omics data. But of course, this is a challenge to really find the connections and to integrate the different types of data with different structures in a meaningful way.
Our project is about understanding the drug resistance mechanism of breast cancer, and we are especially focusing on triple-negative breast cancer because these are the most difficult cases and these are the ones that are most resistant. and we are trying to predict the response to neoadjuvant treatments. So they want to shrink the tumor before doing the surgery. That's why they apply the neoadjuvant treatments. And we are getting different types of omics data, metabolomics, transcriptomics, and the sequencing data from the samples. So we are actually working on clinical data and some retrospective data as well. And we established a database of all this information. And from these, we are trying to build models predicting the response to the neoadjuvant treatment. So in these models, we use combination of different techniques. I'm in charge of the modeling work package. So we are using mechanistic models and machine learning based models, metabolistic network models to predict the response to treatment. And now we have a competition. We are all working on the same data that we decided. And we will see the result of the competition, which model works the best, and how we can incorporate these models to build a better predictor. So that's the task of our work package. And what kind of data we need as an input, what kind of data we can provide as an output. And we also would like to see the incremental change in adding of new information. So how much we can predict from the clinical data only, and when we add the sequence data, how much it improves the model. When we add the transcriptome data, how much it improves the model. So this will give a convincing story to the end users on how much data is needed, what kind of data is needed. And the project will have a high impact on medicine, especially for clinicians, because they don't know a priori the response of the patient to the neoadjuvant treatment. So if you can predict this accurately, they can decide not to give their neoadjuvant treatment to the resistant patients and look for alternatives. And this is a valuable time for patients as well, because in the meantime the tumor can progress, and so you can prevent this by predicting the response a priori. And it will have big impact on the patient's lifespan as well. And if we can show that such modeling approaches can work in breast cancer, we can apply it to different types of cancers and different diseases. And our ultimate goal is to show that AI-based methods combined with clinical information and insights from the clinicians and integration of these modeling approaches can be used in practice. So we would like to show this as an example of how AI-based digital approaches for predicting response to different treatments and diagnostics can be done and can be translated into medicine as well. So this project is a good example of such a goal.
Computational modeling technologies are the key to make the most benefit of genomic and clinical data. The project aims at developing tools that can act as decision helpers in oncology. Computer-generated predictions could improve clinical practice. The main objectives of the ITFOC project include comprehensive molecular assessment for the patient, the use of digital medicine to model effects and side effects of all therapy options, transformation of healthcare practice across Europe by engaging different stakeholders from patients to clinicians, scientists, and politicians. ITFOC Consortium includes a number of 17 partners from 6 European countries. Today is about bringing the ideas and vision of a sustainable ICT-driven approach in oncology by virtual patient modeling techniques. Some of the themes discussed concern the means of establishing strong ITFOC communities, data collecting and sharing across countries, data validation, as well as policy matters, guidelines, and ethical issues. Research will start from studying triple-negative breast cancer tumors, but in the future it could expand to other cancer types.